Welcome everyone, and thank you for joining us today for our webinar titled Using Touchscreen Operant Systems to Study Cognitive Behaviors in Rodents. My name is Martin Hess from Inside Scientific, and I will be your host for today's event. Our session today is sponsored by Lafayette Instrument, also known as Camden Instruments in the UK, and will focus on a translational set of operant conditioning tasks for rodents that have been designed and validated to mimic similar tests found in the clinical research setting. Our speakers today are Julie Gill, Product Manager Europe, and Matt Croxall, Neuroscience Product and Technical Specialist in the United States for Lafayette Instrument. The technology being presented today was conceived by Professor Tim Bussey and Professor Bussey-Saksida approximately 15 years ago while working at the Cambridge University in the UK, and is referred to as the bussey-saksida system. During the registration process, we asked you to submit one question to our presenters about using touchscreen operant systems. We've taken those responses as best we could to put together the presentation content for today's webinar. Today, Julie and Matt will provide an introduction to this intriguing system. They will review the animal test environment and provide an overview of both hardware and software requirements. They will review the broad range of tasks available for neuronal targets, show some of the data obtained during testing, and also talk about integration of the bussey-saksida chamber with other systems such as electrophysiology, optogenetics, and video tracking.
Hello everyone. Thank you for joining us today and for sending your questions. As Martin said, we've received quite a few questions, well over 130, and most of those we will try and answer during this presentation. However, we will be contacting you all by email to answer your individual questions. So if your questions aren't answered here, we will try and get an answer to you. In our webinar today, I'm going to begin our talk by giving a background to the rodent touchscreen chamber development. Then I'm going to hand over to Matt, who will tell you a little bit about our hardware requirements and also tell you something about the ABET2 touch software, which we sell to control the touchscreen chambers. Then I will tell you something about our available validated tasks and also how these translate to validated human tasks. I will talk in detail about the Paired Associate Learning Task, or PAL task. We've received a lot of questions about integrating touchscreens with other measures, so Matt will kick off our question and answer session by talking about these. Many of the animal touchscreen tests have been developed from human touchscreen tasks. About 13 years ago, Cantab was developed. Cantab stands for the Cambridge Neuropsychological Test Automated Battery, and it was first developed for human testing. It consists of a range of tests which were first designed from a knowledge of animal testing. The idea was to develop a range of tasks that would be sensitive to specific areas of the brain. The Cantab tests are all presented on a touchscreen. And to reduce the influence of language and culture, many of the tests involve requiring the subject to touch abstract shapes on the screen. To understand better the human brain and brain diseases, we not only need good human tests, but also animal models that cause the same behavioural defects as seen in humans. To effectively test these animal models, we need good animal tests that mimic human cognitive tests as closely as possible. That is, we require translational tests. This way we can see if the profile of the animal model matches that of the human disease. As the Cantab tasks rely on a response to abstract shapes and are language free, it was felt that they could be adapted for use with animals to produce translational tests. To begin with, some of the human Cantab tasks were adapted to make them suitable for non-human primates, monkey Cantab. The complex human tasks are broken down into their constituent elements, and each element is assessed independently of the next in a step-by-step progression through the test. Guiding the subject through the test in this way restricts the use of alternative strategies and permits an analysis of performance in terms of cognitive components involved. As hoped, these tests have proven to be sensitive to the same specific areas as seen in human tests. As I said before, the human Cantab tests were originally based on work done with animals, and many of these were rodent tests. Work has now gone full circle to develop rodent tests equivalent to human Cantab tests by developing a touchscreen medium for rodents. A lot of this work has been done at Cambridge University by Tim Bussey and Bussey-saksida's group. Again, complex tasks have been broken down into their constituent elements, and the tests have proven to be sensitive to the same brain areas as the human equivalents. Initial work was done by simply bolting a touchscreen onto the end of standard operant chambers, but since then the chamber has been further refined to optimize it for rodent performance. Important considerations when designing translational rodent tasks are a chamber to keep the animal focused on the task, and Matt will be speaking more about this later. We also need effective training in a stepwise progression, which I'll talk in more detail about later. We need full and flexible data to maximize the information obtained from the tasks, and again Matt will cover that and how we do that with our ABET Touch software. We need validated tasks. There are currently 10 validated rodent tasks and over 60 papers detailing these. It's also very important to provide a flexible system that allows ongoing development of tasks so that everyone can easily program their own tasks and then add to the growing list of validated tasks. The touchscreen chambers are an all-round flexible operant chamber. They can be used in place of levers. The touchscreen has successfully been used with the progressive ratio task. They can be used in place of nose pokes. The touchscreen 5-choice serial reaction time task produces equivalent results to the same task performed in the 5-hole box. Also, having images as the stimuli adds a further function over the more traditional stimuli such as lights and tones. Images can be displayed anywhere on the screen, adding further spatial dimensions. And the modular nature of the touchscreens also allows for other elements such as lights, levers, sounds to be added to the chamber. And in addition, video can also be used as a stimuli as well as the images. Additional information can be gained by seamlessly integrating with other cognitive measures. We have had quite a few questions on this, so Matt will talk further about integrating with electrophysiology, video tracking and optogenetics in the question and answer session. So I shall now hand over to Matt to talk about the hardware and the software requirements.
Thank you very much, Julie, for that introduction. As mentioned, I'll be walking us through some of the information on the hardware and software components of the bussey-saksida touchscreen system. We'll start by taking a look at a general overview of the system. You can see from the diagram on the screen that we have a single computer which controls up to 4 chambers or touchscreens. This limitation is primarily due to the computer and number of PCI cards and monitors that can be physically connected to the computer. Each touchscreen is connected directly back to the computer, while the digital I/O, which controls components such as the house light, the reward mechanism, tone, and others, goes through an interface which provides power to the system and an overall on-off switch. The picture on the right-hand side shows a typical 4-chamber system with the 4 chambers mounted in 2x2 configuration on a rolling cart for mobility with all the computer and electronic components mounted on the top. Focusing on the inside of each chamber, we can see the components listed here. This is a mouse version of the chamber, however the rat one is certainly very similar. On the far right side of the image, we have a touch screen connected to this is a set of black polished walls and lid. The trapezoidal shape is something unique to the bussey system, and as Julie mentioned earlier, this helps focus the animal's attention towards the screen and also limits the viewing angle of the monitor. This encourages the animal to view the monitor straight on rather than from the side at a sharp angle, which might skew the images. At the back of the walls is a reward trough or receptacle. This either presents a pellet or liquid for the reward mechanism. This is really strategically located at the rear of the chamber, as most of the touchscreen tasks require the animal to return to this reward trough to initiate the next trial. This means we know exactly where the animal is at the rear of the chamber, facing away from the screen when we present the images for the next trial. Then the animal can then turn around and have a centered, unbiased view of the monitor and stimuli on the touchscreen. We also have the liquid reward bottles for the mouse liquid reward located at the back of the chamber. Rat chambers typically provide a pellet reward, so there's a pellet dispenser mounted at the top, not shown here. We also have some infrared beams that provide some general locomotion activity information, as well as additional IR lighting for the IR observation camera mounted at the top of the chamber. Of course, all this is located on a nice rollout shelf inside of a sound attenuation chamber with the house light, speaker, and previously mentioned camera mounted in the ceiling. Here we have another view of the chamber where we've removed the trapezoidal walls so you can get a full view of the touchscreen. And in addition, the polycarbonate mask that we use in front of the screen. This mask helps to identify the unique response areas and provide additional depth to the touch response to make it a bit similar to a nose poke in a behavioral sense. One important item to note here is that all the mouse masks are located near the lower half of the touchscreen. This is unique because the rats, there are particular paradigms where they require the animal to rear up and respond at the upper half of the monitor. This is accomplished through a shelf added in front of this touchscreen to get the animal to rear up and respond again towards that upper half. Julie will show some pictures that will kind of reference this or show the animal rearing up to respond, and that is strictly unique to the rat chambers. This rearing component was found to help reduce the impulsivity of the rat's response and overall help their accuracy. Lastly, we see the views of the floor, waste pan, and again the infrared beams. During the registration process, we again got a number of questions regarding the touchscreen itself and the type of technology used. While there are a number of different technologies available for touchscreens, from 5-wire resistive, acoustic sound wave, capacitive, we specifically focused on IR touchscreens for the rodent applications, and this is for a number of reasons. First and most importantly is the no force requirement for infrared touchscreens. The animal doesn't need to come in contact with the physical screen and press or exert a force. This not only adds a component to train the animal to actually press or exert a force on there, so you don't have that training time, but even certain mouse strains with motor deficits can't even exert the necessary force required. Without the animal having to come in contact, and in addition to the masks that I previously mentioned in front of the touchscreen, the animal again has more of a nose poke type response, which behaviorally is very easy to train. Also, with certain technologies, such as projective capacitive, which is what's commonly used in all our iPhones and smartphones nowadays, these exert capacitive fields which could influence neural recordings, which we might be doing later on with these touchscreens. And one final note, these newer technologies such as projective capacitive on these smartphones and other technologies provide an overwhelming amount of data, and rightfully so, as we want to interpret swipes, double taps, and other gestures on our smartphones. But that makes it very hard to pick out the intended touch from a rodent. And we've found through testing that a lot of these IR touchscreen technologies are much more reliable and effective with identifying rodent touches. Of course, we do have a small note at the bottom where plenty of researchers are using other technologies besides IR. They do just fine with their research just through our testing, and from a commercial standpoint, we found the IR touchscreens to be much more effective and reliable. I also want to touch on some alternative configurations of these chambers. The main picture here in the center shows a set of modular walls rather than the standard trapezoidal walls on these previous images. This would be utilized if you need some other modular accessories such as levers, lights, or other devices connected to the chambers, which mount in this similar standard square type configuration. And at the bottom we have a number of components for integrating with third-party equipment or other techniques. The increased height reward tray for animals with head caps, a set of walls made specifically for tethered animals with appropriate openings in the lid to pass through the cable, and finally a TTL breakout box which can be connected to the computer and software to trigger third-party equipment or send out event signals to timestamp other data files. Overall, when comparing the touchscreens to standard operant chambers, we have some advantages. The ability to run multiple tasks and experiments which activate different brain regions make them extremely versatile. In addition, in a single session, you can counterbalance both spatial and stimuli within the touchscreens. And finally, the software, which we'll touch on next, provides an easy interface to customize the schedules and images for your specific research. Switching over to the software, the main software component which controls the touchscreen is called ABET2 Touch, produced by Lafayette Instrument. For a quick overview, we have 5 main tabs or sections in the software. These are located on the left-hand side of this table with a brief description of what they do on the right-hand side. Now we'll touch on each of these rather quickly, and while I may go a bit rapidly through this section, please note that if you have any specific questions on any of the features or functions of the software, please don't hesitate to email Julie or I directly to answer any of your questions or concerns. To start, we have the Environment Designer. This allows us to identify what hardware is connected to the software. As ABET was originally designed to handle operant chambers and their various configurations, we can list any inputs or outputs in terms of what accessories are actually attached at the hardware level. In addition, on the left-hand side, you can see our 4 chambers listed, which in a bussey system is standard with that 2x2 configuration. And then finally, in the center middle, we do have some additional settings for the touchscreens, which reference the resolution. Our next tab is Schedule Designer, which acts as our editor. So just like as in Word, you can open up files, make modifications, save, and lock schedules. On the far left, you have a complete list of variables, and they're aptly named due to their involvement in the schedule. In the center area, you kind of have the meat of the schedule, where we're able to change up what the task actually provides to the animal. And then on the right-hand side, we have a code view, which is not actually editable, but is there for any of the programming-minded to be able to view. Our Execution Manager screen lists our 4 chambers on the far left and allows us to then apply the schedules that we just edited or created from the Schedule Designer into each of these 4 chambers. We can enter information such as the maximum number of trials or time that the schedule should be run for, as well as where the data should be saved, and in addition, some session information that we might want to be associated with this particular session. And those are all user-defined, but could be anything from an animal ID to a dose to strain and information like that. We have an Analysis Set Designer tab, which allows you to create custom queries or macros which go through the raw data and pull out pertinent pieces of information. Many of these predefined tasks that Julie will be referring to have these macros or queries already written, but you have the flexibility to open up and add in additional measures that you might want to view. And finally, the data viewer, which allows us to go through everything from the raw data to create daily reports, and really is a primary feature of ABET. We've focused primarily on making sure that at the end of running a day of maybe full of 48 animals, that within a few minutes and a few mouse clicks, that you can easily extract meaningful data to be able to make decisions for testing or training the following day. And just to wrap up in summary, ABET 2Touch has the versatility to control a wide range of hardware, whether it be from Operant to touchscreens. You can also create your own or modify existing schedules. These schedules, you can load in the predefined images from our multimedia library, or you can simply add in your own images that are created in the lab. You also have independent control over each of the chambers, as well as millisecond timing. As mentioned at the data viewer slide, we really focused on being able to quickly view and extract meaningful data to make decisions for the following day. And our guided interface, which offers lots of functions and features and drop-down menus, helps reduce the learning curve while not having to learn a syntax or code mechanism.
So, in this part of the webinar, I'm going to talk about our validated tasks. So we're going to talk about the tasks themselves, their application to different neural areas, typical training times for each task, which rodent strains have been used, and we'll also look in detail at the PAL task. So here are the 10 validated tasks. So we have here a pairwise or visual discrimination task. So here we see the rat task where we have it mounted with the shelf as Matt was talking about earlier, and in the mouse task it would be at the bottom of the screen. In this task, one shape is correct and gives reward, the other is incorrect and gives a timeout. And once the correct shape has been learnt, you can then reverse the task so that the opposite shape becomes the one that's correct. We have the PAL task, where each of 3 shapes are each associated with a different correct position. So you can see 3 positions, and there are actually 3 shapes in the experiment, but you only ever see 2 at one time, one in the correct position, one in the incorrect position. We have the visuomotor conditioning learning, which is a go left or go right task according to the shape that's shown in the center here, which would be one of 2 shapes. We have a location discrimination task where 2 positions are illuminated left and right, and only one side is correct. And once they've learnt which side is correct, you then reverse it so the opposite side becomes correct. The tunnel task is a non-matched position task which uses 15 locations in the rat and 5 locations, just 5 along the bottom, for the mouse. To start with, a sample position is lit, and that's followed by the same sample along with a new position being lit, and the correct response is then to the new position. And because that new position can often be either side or below or above the sample one, the rodent cannot use mediating strategies to help them remember, making this a working memory task. The 5-choice serial reaction time task is the same as performed in the 5 or 9-hole boxes, where the rodents have to respond to a briefly shown image shown in one of 5 positions. Auto-shaping task is not actually shown here, but for the auto-shaping task we move the reward tray so that it's in front of the screen, and then we light either left or right of the reward tray. When one side is lit, that'll be followed by reward being delivered, and when the other side is lit, then no reward follows. And what you tend to find is that the rodents develop a Pavlovian response towards the side of the screen that they associate with food being delivered. There's an extinction task where you simply reward for touching a stimuli in the center, and then after a while you stop rewarding and see how quickly the animal gives up and touching the screen. The progressive ratio task, in place of the usual lever for this task, we have a white square shown which disappears briefly on being touched. The mouse is required to touch that stimuli progressively more times to get reward. Initial work indicates that the touchscreen PR task produces less variable data and is more sensitive to manipulation which promotes PR performance than the equivalent lever pressing task. And one of our newest tasks is the rodent continuous performance task, where 5 shapes are briefly shown on the screen in a random order. But only one of those shapes will give reward when touched, and the rodent has to learn to withhold responses from the other 4 shapes. We also provide a 5-choice CPT task and a 4-choice gambling task, though these are not currently validated in the touchscreen, but simply based on tasks done in the 5 or 9-hole boxes. As mentioned before, the tasks are designed to target specific regions of the brain, enabling a battery of tests to be compiled for different research areas. This table shows which neural systems are involved and which clinical areas show deficits in the task. So we can see that the pairwise or visual discrimination and visual motor conditioning task target the frontal brain areas, therefore are useful in Huntington's and Parkinson's disease. Whereas the PAL task, the location discrimination task, and the tunnel task activate the hippocampus and other areas associated with memory, and they are very good for Alzheimer's disease. A lot of these tasks, you will see, are also useful for schizophrenia studies. Here we have a range of attentional and motivational tasks, and which adds to the dimension a range of cognitive functions that can be measured and tested, and associated disease areas, which include ADHD and OCD. This table shows typical times taken to train the rodents on each task. Times vary from a few sessions in the auto-shaping task, as shown on this slide, to 35 to 45 sessions for the PAL task. Many of the tasks have the same initial training paradigms to train the animals to touch an image and initiate a task by nose poking the reward tray at the back of the chamber. This typically takes 1 to 2 weeks, but once you've trained for one task, you don't need to repeat the whole procedure for the other tasks. You will see that on the whole, training times are very similar for both rats and mice. So here we see training times for our other tasks. You will see that we have a missing point here for the progressive ratio. That's simply because that hasn't actually been validated in the rat yet, but we hope to have information about that soon. Here are a list of the rat strains that have been reported in the literature as being used with the touchscreen chambers. There are probably more strains that have been used, and we're currently asking customers to let us know which rodent strains they have used and how they perform. So if you're listening today and can help us with that, we'd love to hear from you. Even the lines with weaker vision, such as the Sprague-Dawley rats, perform well on the touchscreen tasks. Dr. John Howland of Saskatchewan University has used genetically modified epilepsy rats with a Wistar background and showed them to be deficit on the PD or visual discrimination task compared to their non-epileptic controls. Several strains of mice have also been used with the touchscreens, including transgenic and knockout mice. We will look at one of these strains' performance in the PAL task later. We will now see, we will now address the translational nature of the tasks. Here we see a list of the tasks which have equivalent versions in monkeys and human touchscreen tasks. The visual discrimination, continuous performance and 5-choice tasks all have very similar tasks across the species. The spatial working memory task is the tunnel task in rodents and a spatial ordered search task in monkeys. And the human task is actually called spatial working memory, and that is a more complex spatial ordered search. But all of them rely on remembering which positions have already been visited and not revisiting those positions. The set shifting task is yet to be validated in rodents, so we hope to be able to have a positive tick for that in the future, but it is available for monkeys. This graph just demonstrates how different disease areas have a different profile across the tests. It shows the results for mild Parkinson's and mild Alzheimer's patients. Comparing the PAL and set-shifting IDED tasks, we can see that Alzheimer's patients are deficient on the PAL task, but are okay on the IDED, whereas the Parkinson's patients show the reverse. We will now look in detail at the PAL task. This is a task that correlates to learning and memory and is highly sensitive to Alzheimer's disease. In the human task, 6 boxes open one at a time in a random order. And behind each one is shown briefly a random shape. Each shape is then shown in a random order in the center of the screen, and the subject is required to touch the box where they first saw the shape. If a mistake is made in any of the shapes, the 6 boxes are all opened again to show the same shapes, and the task is thus repeated. This makes it a learning memory task. Most people will learn to remember all 6 shapes after being shown them 2 or 3 times, but this ability to learn to remember is what goes very early in Alzheimer's disease. Here we see some work done by Kylie Fowler in Australia. She tested mild Alzheimer's group, a group that reported some memory problems, which are labelled here QD for questionable dementia, and a control group. And she tested these on the PAL task over 2 years. The Alzheimer's patients made more errors than the other groups and got steadily worse over the 2 years. The control group made few errors and their performance remained steady over the 2 years. The questionable dementia group, however, split into 2. One in green, which performed like the controls, and one in red here, which followed a line similar to the Alzheimer's patients. And at the end of 2 years, all were tested on cognitive tests then used to diagnose Alzheimer's disease. And all of the subjects in red were diagnosed as having Alzheimer's disease at that 2-year period. And this slide just shows you the individual data for that questionable dementia group, so you can see just how tight data is across the subjects. And the 34 error cutoff, shown in red here, would have identified at the 6-month period those subjects who would go on to receive an AD diagnosis. Here we see results from a study done by Barbara Saharkin's group in the UK. They used the PAL to distinguish between mild Alzheimer's patients and patients exhibiting memory problems through depression. They also did a study with a questionable dementia group and of course had a control group as well. And like the previous Fowler study, they found that the questionable dementia group could split into 2 and that all of these above this dotted line here went on to get Alzheimer's disease. So here we see the monkey version of the test. In the monkey version, 4 shapes, this is at the hardest version, 4 shapes are shown one after the other. Each shape is in a different position on the screen. And after that, again in a random order, 4 of the same shape are shown, and the monkey has to point to the shape where they first saw it on the screen. So here we see the monkey doing that task at the very earliest stage. So in this case, it's just 1 image and 2 positions. So it starts off with a sample being shown, which he touches, and then 2 of the same are shown, and they have to touch the one that was shown before, touched previously. So that's the very early training part of the stage, but rhesus monkeys can readily acquire this task at the 4 stimuli region. And as with humans, the more time they see the location, the better their performance. Ketamine and scopolamine both disrupt performance. Ketamine-treated monkeys require additional attempts to learn the task, but scopolamine-treated monkeys do not learn the task after being shown it repeatedly, just as we see in Alzheimer's patients. It therefore appears that PAL can distinguish between a lack of sustained attention and impaired learning memory. Here we see the rodent task. There are 3 positions which are marked out by placing a mask on the screen. Each trial has one shape which is in the correct position and one shape which is in the incorrect position. So you can see here the 6 different possible trials that can be presented. And here we see a mouse doing the task. So he initiates the task by going to the rear and then touches the screen, and because he got that correct, it's now taking the reward. Then we go into an inter-trial interval, which is around 20 seconds normally. After that period, the light will light up in the reward area, which is at the rear, and he touches it and another image comes up and goes for another reward. So here you can see a very well-trained mouse. So training on the PAL and also many of the other tasks consists of several stages. So after getting the animal used to being handled and then food depriving the animals, we habituate them to the chamber for about 15 or 20 minutes. After that, they go on to a Pavlovian training stage where images are shown on the screen, after which reward is given at the rear of the chamber. And if the images are touched, there is immediate reward. Next, the animals are trained to touch the images by leaving the image on the screen until they are touched, when a reward is given. Then the animals are trained to initiate a trial where they have to enter the food trough at the rear in order to bring up the image, which they can then touch to get the stimuli. Getting the animal to the back of the chamber, as Matt mentioned before, is so that they can see all of the images before responding. Finally, they are trained to ignore the blank windows when the images are shown by punishing the touches to the blank windows with a timeout period. And this saves time during the task proper, although responses to blank windows are not punished during the actual task. The animal is then ready to move on to the task. The tasks already described are called Deepal because they use 2 different images, or there are 3 different images. 2 different images are shown at the same time on the screen. There is also a version called SPAL, where we use the same image, one in the right position and one in the wrong position. Antagonism of NMDA and AMPA receptors impairs performance on this Deepal, but not on the SPAL. And this is believed because the SPAL can be solved by a go right, go left strategy, rather than remembering precise positions. Humans with a DLG2 mutation have been found to be impaired on PAL, and likewise DLG2 knockout mice also show an impairment, as shown here when we compare the wild-type mice with the DLG2 mice. In summary, there are a wide range of validated tasks covering a wide range of cognitive functions. Training on the task varies from a few days to 1 to 2 months. And the chambers have been used with a wide range of rat and mouse strains. Touchscreens offer an alternative to standard lever and nose poke responses. So I will now hand over and we will go to our question and answer session.
Matt, during the registration process and throughout this presentation, people have asked about integration with other technologies. And I understand that you've prepared a few extra slides just to provide a bit more detail about integrating the bussey-saksida chamber with other equipment. So I'll let you just talk a little bit more about that.
Yes, Martin, absolutely. It's certainly a question that comes up quite often, and there's quite a bit of information that we could provide on this. We could actually probably do a whole other seminar just on integrative approaches. But I've tried to summarize a few points here specific to electrophysiology and optogenetics. For electrophysiology integration, we do have chambers that are compatible with third-party neural acquisition systems from any of the main neural companies. These chambers have some specific features which make the recording of neural data a lot easier. Starting with the sound attenuation chamber, it's going to be lined with either stainless steel or a metal paint, which will help reduce emissions from outside the chamber getting to inside where we're recording. Also, the touchscreen itself, which has a small source of EMI, has an additional film over the monitor so that we reduce the emissions from the monitor to the animal. Also, the electrical components from the pump or pellet dispenser motor to any other cabling has been additionally shielded and grounded to, again, reduce further emissions from inside the touchscreen. And as mentioned previously, TTL lines to send event markers to the acquisition system to mark events in your neural file are provided as well. So we have a picture of one of those chambers here mounted to the left. For optogenetics and video tracking, we again have some components that make the integration to these other systems a lot easier. Previously mentioned, we have these reward areas with increased height, as the standard reward areas have a much smaller entry. And so for any animals with a head cap, this larger opening is immensely important to be able to get access, easy access to the reward. And as we always mention, these TTL output trigger lines to trigger a laser or send event markers. So we have a picture of our TTL breakout box, which connects back to the computer and the ABET software. And then additionally, the walls that have been designed for openings in the top for tethers and cables. And we also have a rolling shelf towards the top of the chamber, which we haven't been able to show a picture of, but for easy mounting of commutators and other devices up above the actual enclosure with the walls and the lid. Now, Lafayette and Camden have been working on a fully integrated solution, however, that is designed off an acquisition system that we've created that is specifically tailored towards neural data acquisition inside these touchscreen chambers. Many of the other acquisition systems don't necessarily have the behavior chambers in mind when a lot of their components are created. Therefore, it's really difficult to cram those into a very space-tight touchscreen chamber. So we've taken it upon ourselves to try to make that process a bit easier and a bit off the shelf. So these new chambers would contain all the same features of these electrophysiology compatible chambers, the shielding, the additional grounding, the film over the touchscreen, but we also have some additional components that would, again, benefit specifically inside these touchscreen chambers. One would be the auto retract tether, which is a commutator with an auto retract feature to take up any additional slack of the cabling. Also, the head stages are, of course, all digital. And when that gets passed back to the acquisition system, we are synchronizing and providing data streams of both the behavior, the neural data, as well as video, again, all synchronized together off one clock. And of course, these are all designed, again, to be specifically used inside the mouse or rat version of the touchscreens. And we have both a tethered option, primarily shown here and on the previous slide, as well as a wireless option as well. So while, again, there's plenty of information that we could go on and on about regarding these integrated solutions for electrophysiology, for optogenetics, video tracking, for microdialysis, or other applications, we could spend a lot of time here. Please email any questions to either Julie or myself, and we'll be able to either point you in the direction of someone who's done this integration or, again, help in terms of listing out components that might be helpful in that particular scenario.
Great. Thanks, Matt. Thanks for that additional information. I'm sure that was really helpful. Actually, we did receive a couple comments as you were speaking about wireless integration with wireless head stages and things like that. So I think you've answered that well. And as it relates to the software, are you able to provide our audience with a very quick software demonstration to show what the data looks like when it comes out of the system? There were quite a few questions about that as well.
Sure, we could pull up ABET here and take a quick look. We have, again, referring back to the software section of the presentation, we have our 5 tabs here listed above. I'm over here in the Data Viewer tab, which allows us to search through our database and find particular sessions run on particular days and in specific chambers. I'm going to go ahead here and just highlight 3 sessions of data run back on January of last year. And initially on the right-hand side, I get some information that's just general session information, but you can see here I have plenty of other tabs that will provide me different views of the data. One unique feature of this system is that we record every piece of data in a time series. So it doesn't matter if the IR beam break is pertinent to that schedule. It doesn't have to progress the schedule, or the touch to a screen doesn't have to actually mean something in the schedule. It's still recorded in the data. The one benefit of this is after the experiment is all done and you're after a latency or a time measure, and even if you didn't program a timer in or a variable to keep track of a particular count or a latency, all that information is still there, and through that analysis set designer you can still pull that information out. But going back here, I've selected 3 sessions. I'm going to load these into a center column where I can select them again, and it'll sort them for me. And then this analysis tab, which is by far the most utilized data tab, allows me to run these pre-written macros. I've selected an analysis folder for the pairwise discrimination task, which, as Julie described earlier, is basically a discrimination between an S plus and an S minus stimulus. And then I get to select an appropriate analysis macro for the schedule that we ran. This is a final version of the task, so at the very end of the training. And once I hit execute, it goes through and processes those 3 sessions that I have selected. Now this first screen gives me some group statistics, but what the real benefit of ABET is this report editor button that I can click on and kind of design my own spreadsheet of how I want to view the data. On the left-hand column here, I have session information such as the animal ID, the date, what chamber, and any of the session information that I entered in the execution manager can be added here at this time. And it's easy as just selecting that, and it'll appropriately add that information into the spreadsheet below. There's a duplicate. And in the center column, I have my measures that are pulled out. So everything from the number of trials completed to the percent correct to down here, I have my latencies. Now what's unique with the latencies is that I can add a detailed view which will give me a column for each value that it has. So if the animal went through 30 correct touch latencies, it's going to give me a column with each value, which is a bit more data than I'm actually after. So I'm going to remove that, and by adding an aggregation here like my mean and an appropriate label, I can easily get at maybe an average latency for each session. In addition, this whole template can be saved, and I actually have a previously utilized report template that has all that data already pulled up and in the format I want, and from this screen I can easily copy that to the clipboard or export it to a file like CSV and paste it into Excel and keep track of this on a daily basis. But this would be information that I can make decisions on for tomorrow's training. Here I can see that my percent correct for these three animals was 86, 93, and 96, and they all got through 30 trials. So again, these animals were well adept at this task. This guy might need some additional training, but again, we're at very good stable levels. Okay, and I again ran this for 3 sessions, but you could expand this over any n value that you wanted for today or whatever sessions you were looking at. So again, there's plenty of other features through ABET. If there's any questions or if you'd like additional information, please don't hesitate to contact us.
Great. Thanks, Matt. Okay, I'm just going to sift through some of the questions here. I know it has been an hour, but we're going to keep the session open for another 5 or 10 minutes just so we can get to some of these additional questions. So, Julie, I'll start by asking you this question from Yuichi at Harvard University. And the question is, how do you compare the speed of training for your 5 CSRT task to the same task implemented in a traditional nose poke setup? And part 2 is nose poking the touch screen as intuitive to mice as the traditional nose poking?
The response on the touchscreen is very, very similar to training to nose pokes. So it's neither quicker nor shorter. And I think that's probably because it's to the mouse, it's very similar to nose poking. By putting these masks in front of the screen and having this infrared, very, very sensitive infrared technology, the animals really only have to kind of nose poke into the area provided by the mask. And so from that point of view, the nose poking and so the training basically on the touchscreen is very similar to that in the nose poking boxes.
Okay, fantastic. And actually, that's a great answer, and maybe this will segue into another question, perhaps for you, Matt. Maria has asked, she's just asked for you to elaborate on how the touch is actually detected by the infrared sensors. So can you just elaborate on that a little bit more?
Absolutely. The IR touchscreens themselves have kind of an infrared matrix over the top of the screen. So they have a number of beams in the X and Y direction, similar to kind of how old open field frames detected where the animal was located in an XY coordinate system. However, with the IR technology, over the years they've advanced that, so your resolution in terms of where the touch is isn't just the number of beams in the X and Y directions. Via triangulation of looking at the reflection and absorption of the IR beams along each path and pulsing them in a sequence, they can actually give a very, very fine resolution of where the touch is. So that's sort of how the technology works. There's a very thin bezel right above the glass of the monitor, which contains all these beams. That's a very important cleaning area that needs to be kept very, very clean. But really, we only see the bottom bezel area get a lot of dust and dander while running these chambers.
Okay, perfect. And just for clarification, so really all the rodent needs to do is get their nose within, say, a millimeter or something like that of the screen, and you know, it's registered as a touch.
That's correct.
Okay. Julie, question for you from Nick also at Harvard. He's asking if there's a task for compulsion, for example, like the DRL-72 test.
I'm not familiar with that test off the top of my head, but for the task for compulsion, we've got the extinction task, so we can see you train them to respond and then see if they stop responding when you no longer give them reward. And also the CPT task that I was talking about, where we're flashing images in front of them and they have to actually withhold a response to 4 out of 5 of those images. So that means controlling that sort of compulsory element quite finely.
Perfect. Okay, another question about, let's see here. So I'm just sifting through. We've had this question a couple times here now, and Just from the Rutgers Brain Health Institute in New Jersey has asked if we can train the rats to hold a screen touch for, you know, several seconds before releasing the screen touch. Can either of you comment on that?
I think you've got some experience with that, haven't you, Matt?
Yeah, we've had a number of customers who have added that requirement to their tasks for specific experiments where they've required the animal to dwell, just like in a nose poke, for a certain duration. And the software does detect the onset of the touch and the touch up event, or the release of the touch. So in the schedule, you could clearly put in a requirement for the animal to make the initial touch and then stay there or dwell for a variable amount of time before they have to release. So that is certainly doable.
Perfect. And maybe this leads into the next question too about the pre-programmed or canned tasks versus one that one can program for themselves. So I assume that the canned tasks can be modified for difficulty level, for example.
Yeah, absolutely. The whole thing is open, and because it's quite a user-friendly programming way of doing things, it's quite easy to go in there and change things. But we also provide training as well, so people can come to us for additional training on how to program the system if they want sort of a fast track rather than reading through the manual.
And many of these pre-canned tasks come with some common manipulations to increase or decrease difficulty of the task.
Yeah, a lot of those can be changed just at the execution manager stage as well, so you can change them just prior to running the task, as well as going in and changing them in the task itself.
Okay, perfect. Moving along, quite a few people have asked about the age range of mice, for example, that can be used with a system like this. Can either or both of you comment? Is there a minimum, maximum?
I'm not sure of actual sizes, but I know that quite young animals have been used, and certain studies have looked at aging mice and rats as well. So I don't have that data off the top of my head, but I believe it is there in the literature.
Okay.
We can certainly send out bibliographies to everybody. Perfect.
And actually then, Julie, on that note, let's let's see here. I'm just trying to find this question. You had presented a couple of slides with all the neuronal targets and the different tests and what they test, which deficits. So Lorenzo's asked if you could provide references for those particular tests. I assume you have something like that available.
Yes, we have our bibliography. We can certainly send that on to all the registrants, okay. And that data just came straight out of the bibliography.
Perfect. All right. Let's see, maybe we will ask one more question, and then as we've said before, we will make sure that all these questions are answered through our Q&A report, and you will also be contacted with answers to your questions. So let's see here. Are there any sort of specific use and care instructions when it comes to either cleaning or also, avoiding damage to the equipment by the animal itself? Can either of you talk about that?
Yeah. Do you want to take that one, Matt?
Certainly. The cleaning, most of the components come apart, and so it's built in a modular fashion to make it as easy to clean as possible. Most components are going to be either stainless steel or polycarbonate, so anything less than a 70% ethanol solution will be just fine. We have plenty of other labs that use kind of like a Virox or a non-alcohol-based cleaner. And the touchscreen itself is glass, so it's very difficult to harm that. And the animals really don't abuse much of the equipment. They'll obviously chew in certain areas if you leave them in there or don't feed them, but there's not really too much abuse that the chambers themselves will take from the animals. It's usually due to low cleaning frequency that you see a bit buildup of foreign material on the chambers and maybe cause an IR beam or two not to work. So that's typically the source of any sort of issues with the equipment is just a low cleaning frequency.